Today we're going to talk about meiosis. Meiosis is the division that happens in your gametes. So if you think about it, you have 46 chromosomes, but you give half of them to your offspring, or your parents gave half of them to you. So if your cells that went into creating you just did mitosis from your parents, then you would have 92 chromosomes, and that's not going to work. So we need something to reduce the number of chromosomes in our gametes, or sex cells, or sperm or egg, or our reproductive cells, whatever you want to call them. This involves two cell divisions. It's kind of like going through mitosis twice, but it's not exactly. And it produces four haploid cells. So this is a new word for us right here, haploid. Your normal cells are diploid. You have two copies of every chromosome. A haploid cell has one copy of every chromosome. So if I give a haploid cell from your mom and a haploid cell from your dad, I get a diploid cell that makes you that has all of your chromosomes. If we look at the steps are named the same, but they have Roman numerals after them. Okay? So here we have interphase and G and S duplicating the chromosomes. So that's normal. But then you go into meiosis. And we start with a prophase 1. Okay? Notice in a prophase 1 there's crossing over. Crossing over continues into metaphase 1. Crossing over is very important because it adds to genetic variability. In metaphase 1, here's the biggest difference between mitosis and meiosis. Instead of lining up as one giant line, as in mitosis, in meiosis, in metaphase, they line up in what are called homologous pairs or homologous chromosomes. Chromosomes that have the same genes. So your chromosome 1 from your mom and your chromosome 1 from your dad. Your chromosome 17 from your dad and your chromosome 17 from your mom. They line up in those pairs. And then the next change in meiosis is in anaphase 1. In anaphase 1, instead of separating the chromosomes like you did in mitosis, it just moves the chromosomes to the other side. Notice they're still duplicated chromosomes, but they've just moved. Okay? Also notice that this blue one has a little orange and this orange one has a little blue. That's a crossing over event. They've exchanged genetic material to make the offspring slightly more different. Then you have telophase 1. Okay? Similar things happen. Nuclear envelope reforms around the chromosomes. The centrioles break down, or spindle fibers break down. The centrioles are there. But notice that the chromosomes are still duplicated. Right? We have cut the number of chromosomes in half. That's a key. We had to do that to begin with. We started with 4 chromosomes. Now we're going to have 2 in each cell. Okay? But they're still duplicated. Meiosis 2, prophase, metaphase, anaphase, telophase 2, is very similar to mitosis. Same things happen in prophase. Nuclear envelope breaks down. Spindle fibers start to form. Centrioles start to migrate to the poles. Metaphase 2, they line up in one single line, just like in mitosis. Anaphase 2, the chromosomes break apart, just like in mitosis. And telophase 2, similar things happen. Cell starts pinching, nuclear envelope starts forming, chromosomes start unraveling, centrioles just sit there because the spindle fibers are broken down. And then you have cytokinesis, which breaks the cell into 4 new cells. Notice I got 4 cells. Okay? And notice if you look very closely, each of these cells are slightly different. This one, the blue is continuous. This one, the blue has a little orange. This one's the orange has a little blue, and this one, the orange is continuous. Okay? It adds to the genetic variability of the offspring. Also, these are your gametes. So if this was a male, these would be sperm. If this was a female, they would be eggs. And notice this little line. If this was your father's, you would get a cell from your mother, and they would become the zygote, the first single-celled you that has 4 genes, just like the original cell had. And you would have 46. Last thing, crossing over. Crossing over occurs in prophase 1 and metaphase 1. It's very important. It's when homologous chromosomes, hey, that's a vocab term, homologous chromosomes, chromosomes that are the same, exchange parts. Now, when I say they're the same, I mean they have the same they have the same genes. So they both code for hair color, they both code for height, they both code for hitchhiker's thumb. But they might have different alleles. One might code for hitchhiker's thumb, one might code for straight, but they're both coding for thumb shape. Okay? So they exchange alleles of those same genes, and this creates more variability in the offspring, which makes nice little fuel for evolution. All right, thank you, and that should be the last video lecture for this chapter.